Hi, guys. Welcome to our second screencast this week, which is going to be about the cell cycle and mitosis, which is essentially cell reproduction. I thought we could start today with a little interactive animation. Let me hide this so that you can just get an idea for what this looks like before we talk about it. And then we'll come back to this at the end and look at it again. Okay, let's try it again. This is taking you through all of the stages of animal cell mitosis. I like to look at it this way because when we look at it in different sections, it does seem very separate, but in fact, it's one long, smooth process. Okay, so let's start. So we're going to start by thinking about the cell cycle, which is essentially the life cycle of a cell. Nothing lives forever. Cells go through all of their stages and eventually will reproduce and go into two cells. Now we have the time when a cell is not dividing, when it's just going through its regular cell life. Excuse me, that is called interphase. And if you look at this, this is pretty representative of that. It's about 90% of a cell's life, interphase. That phase is divided into three subphases, which we'll talk about. And then we have what's known as the mitotic phase, which is cell division, mitosis, and then cytokinesis. And we'll go through that too. Okay, so interphase is when a cell is just living its regular life. It's performing all of its cellular duties. It's making molecules, it's making organelles, it's doing whatever it needs to do to perform all of its functions. And like I said, there are three um, phases of this: G1, S, G2. And we're going to go through all of these separately. Okay, here's just a little thing about interphase, telling you again what that means. The cell is growing, it is replicating DNA to prepare for division, making sure that it's all good to go. And if you remember from our discussion on chromosomes, at this point, DNA is chromatin, like that big bowl of spaghetti. Okay, so let's start with G1. G1 you can think about as gap one, you can think about as growth one. This is immediately after a cell is born, essentially, right after cell division. Um, the cells are about half the size of the original cell, so they're baby cells. During this phase, the cells will grow to mature size, meaning they're just going to get as big as they need to get. Once the cells are grown and mature, that will also include making enough organelles that the cell will need. So let's say it's a muscle cell, it's going to need a lot of mitochondria. It's going to make all of those organelles. Once the cell is all grown up and mature, it will go to the next phase if it gets the correct signals. The next part is S. S stands for synthesis. And remember, to synthesize means to make. So at this point, the cell is replicating all of its DNA. We will actually cover that in another unit. So we're not going to go too much into that now, but just keep it in mind. DNA is duplicated. It is still in the form of chromatin, so it has not condensed into chromosomes yet, and it's preparing for cell division. Okay, the last part of interphase, G2, you can think of it as gap 2. You could think of it as growth 2. This happens right after DNA synthesis. And basically at this point, the cell is getting ready to divide. So it might have to grow a little more. It needs to make sure it has enough organelles to split between the two new baby cells that are going to be born. And it will also check on its DNA to make sure that everything was replicated perfectly. Okay, so now at this point, after G1, S, and G2, the cell is ready to go into the mitotic phase, which means it's going to divide. And what we're going to do is go through all of these steps one at a time, just like the animation that we watched in the beginning, and then we'll come back to that animation to see again what that looks like. Okay, here's just another diagram of the cell cycle, again showing you that most of the time a cell is in interphase. About 10% of the time, a cell is in its mitotic phase. Excuse me. Here is a nice diagram showing you what this process looks like, what each stage is, what's happening to the chromosomes. Because again, one of the main things that's happening here is the duplication and then pulling apart of those chromosomes so that every cell has exactly 46 perfect identical chromosomes. Okay, our first stage is called prophase. Um, at this point, your chromosomes will start to coil up around those proteins and condense into chromosomes. So chromatin will condense into chromosomes. A structure called a centrosome starts to move to the opposite poles. Think about the poles like the North Pole and the South Pole, right? These structures, centrosomes, are going to help pull the chromosomes apart towards the end of the process, as are these spindle fibers. Spindle fibers will be connected to the centrosomes and then eventually the chromosomes, and they will help to pull everything apart. Now, these spindle fibers, if you think back to when we did our cell unit and we talked about microtubules and cytoskeleton and all that stuff, these spindle fibers are microtubules, meaning they are protein-based structures, and they only appear during times of cell division. In general, they are not present when a cell is just living its life. Next is prometaphase. Some resources don't include prometaphase, they just go right from prophase to metaphase. It is a transition period between prophase and metaphase. Sometimes it can be referred to as late prophase, but some really important things happen here. So either prometaphase or late prophase. Your nuclear membrane breaks down, right? You have to set all those chromosomes free so that eventually they can move to opposite sides of the cell and become their own cell. The centrosomes are at the opposite poles, so one at the North Pole, one at the South Pole. And then those microtubules will actually capture the chromosomes here. So if you remember when we talked about chromosomes, we talked about centromeres, which are like a belt that holds the two chromosomes together. That belt will also attach to the spindle fibers. And that is one of the ways in which these chromosomes will eventually be pulled apart to make two new cells. Okay, metaphase. Meta in general refers to middle. So this is the middle phase of mitosis. And what's happening here is things start positioning themselves so that they can be split in half. Now, this is the phase where your chromosomes are most visible and where people will capture micrographs, which is a photograph taken through a microscope of those chromosomes to make a karyotype. They are well defined, they're very condensed, and they're basically lined up in such a way that you can see the pairs. So that's kind of a nice thing to think about. The chromosomes, and this is such a nice visualization, will move to the center of the dividing cell so that eventually they can get split apart into two. And you'll see here, right? This is your chromosome. This is your centromere. One chromosome is here, one half of the X, one half of the X is here, and they're getting ready to be separated. Anaphase, a nice way to think about anaphase, because it really is separation of the chromosomes. A for anaphase, A for away. And you can see here, this is a nice visualization as well. These sets of chromosomes are being pulled apart. So those are sister chromatids, meaning identical chromosomes attached to each other. They get separated, and move towards the poles of the cell. So now once these guys are separated, they're no longer sister chromatids, they are separate chromosomes. And that is something that I feel like people get stuck on a lot. The idea that you can have a chromosome that is an X, that is two sister chromatids attached, and you can have a chromosome that is one single chromatid. They're both chromosomes. One is sister chromatids, one is a single separate chromosome. Okay, telophase. Telophase starts as soon as the chromosomes start to arrive at the poles of the cell. So you can see at this point, it basically looks like you have two separate cells. The spindle fibers that pulled the chromosomes apart start to come apart. So those proteins that made them will just go off to be recycled, made into something else. Chromosomes uncoil and go back to the chromatin state. So we go from those nice condensed lines to big bowl of spaghetti. And then you also get the reforming of the nuclear envelope around each new set of chromosomes. So those chromosomes will then be captured inside a nucleus. A nucleolus will also form so that you can still have the production of ribosomes. And this is just about two separate cells. The last part is called cytokinesis. So at the end of telophase, the cytoplasm and the cell membrane starts pinching inward, forms what's called a cleavage furrow. And this will eventually pinch off, the membrane will reform, and you end up with two complete new identical cells. And that's an important thing to note. The goal of mitosis is to give you two new identical cells, identical to each other and identical to the starting cell. That is very different from meiosis. So it's an important point to make because we're going to talk about meiosis next. Okay, one last thing for us to talk about are checkpoints. So, all along this process, your body, your cells have checkpoints to make sure that this process is happening correctly. Because if this process happens incorrectly, you can run into big, big trouble. Cell division needs to be a controlled process. And when things get out of control, that's when you can have things like tumors and cancers and things that the body has a very hard time regulating. So, we have three checkpoints. G1 happens here in your G1 phase. Really, what it's looking for here is that the cell has grown enough because it really needs to double in size from the time that it's born to the time that it starts preparing to divide again. It needs to double. So, G1 checkpoint, is it big enough? G2 checkpoint happens at the end of G2, which is after the S phase. This checkpoint is really going to make sure that the DNA replicated properly. What that means is every chromosome needs to now be two identical and perfect sister chromatids. If your DNA does not replicate properly, your new cells won't be right, and that can again lead to lots of problems. And then lastly is your M checkpoint. So once the cell divides, did the cell divide properly? Does each cell have exact identical 46 chromosomes? Does each cell have the organelles that it needs? Are all of the membranes right? Did the nuclear membrane reform? So your cells are going to be looking for all of this damage. If damage is found, if something doesn't happen properly, things can be repaired. The cell cycle can be stalled until repairs are made. DNA can be repaired, membranes can be repaired, whatever it is that needs to happen. If the cell cannot be repaired, it's targeted for destruction. So it'll get tagged with a protein, and then your immune system will come along and get that cell and keep it from replicating because this process really does need to happen perfectly. And again, uncontrolled cell division can lead to things like cancers, tumors, all of that. Okay, I feel like I went through that so fast, but it's so hard not getting any feedback. Let's go back here and watch this again, right? So here's all of your phases. Okay. See how you go from X's to singles as those are getting pulled apart? I like that. Okay, now this is a Cells Alive website. There's a lot of good information on here. I'm not going to assign you guys anything from here because I feel like I gave you a lot of work this week. But, if you're looking for stuff to do or you need a little more information, this might be a good spot. All right, guys, thanks so much for this screencast. We'll be checking in this week to make sure you don't have any questions, and I'll talk to you soon.